let's keep the thought process going here on technology and science with Fanny Elahi. She's a physician scientist, leads a science lab at the Mount Sinai Hospital School in New York City that is rethinking Alzheimer's, among other things. Earlier diagnosis, a huge breakthrough in this scourge, and here today to update us on the major science research on Alzheimer's and dementia. Please welcome to our stage Fanny Elahi.
Hi everyone. It's a pleasure to be here, and I think my topic is not the happiest of topics, but I'll try to tell you why I'm so excited to be working on dementia in this decade and why there is great hope to come. The choice of wording, ending, is on purpose. I can't say cure dementia. That would be very misleading because it would mean that dementia is a disease that we can cure, and it's not. However, how we need to end dementia is by detecting early, because there will be a start to the degenerative process of the brain and the dysfunction of the brain that eventually, when gone on, untreated, undetected, will lead one person to lose their functions, lose their identity, and develop dementia. So dementia is a clinical state akin to heart failure from many different causes, or metastatic cancer from any kind of source. What we need to do to end dementia is to detect disease, any number of diseases that lead to dementia early, while function is unimpaired, and to understand what we need to treat. We don't need to treat everything, but there are ways to understand what we need to go after. And then to be able to do this at scale. If we all are lucky enough to live long enough, we will develop brain degenerative diseases. We can do nothing about brain aging. So if people are dying before they develop dementia, it's because they have died of other causes. Therefore, this is a problem for every single human being living today. Now, when I say change, what are we thinking about? Memory impairment, inability to perform our daily task, inability to speak or to understand language is how we've come to define various kinds of syndromes or presentations of dementia. And yet, how brain dysfunction begins is with changes in mood, sleep, movement, our sense of connectedness to one another, and yes, cognition. But often, it's not actually the first changes, it's just the first that we note. And in fact, maybe we've come to recognize dementia as Alzheimer's disease because memory impairment is how most people come to the medical attention. What my lab is really interested in, and how I think we're going to conquer dementia and end it, is by detecting molecules associated with brain degeneration and dementia. Because that we can screen. We don't need to wait for function to have declined, we can screen. Dr. Alois Alzheimer's at the turn of the 20th century described the very first case of Alzheimer's disease, Augustin Dieter. Interestingly enough, she did not have your typical age-associated Alzheimer's disease or senile dementia. She was 51 at that time, and she had full-blown dementia. So dementia is different than aging. The two are separate. Age is a risk factor for dementia, but the two are not the same. And in fact, although she harbored the hallmarks of Alzheimer's disease, she had a lot of other pathologies in her brain, not just Alzheimer's disease. Eventually what happens is that the brain degenerates, and as the brain degenerates, symptoms develop. We're doing it upside down. We start with the symptoms. Someone comes to my clinic to see me as a patient, and they have full-blown symptoms. That's too late. However, by studying people who have been symptomatic and have donated their brain to research, we know that there are two things that happen. No matter how the brain gets there, eventually, if someone has dementia due to Alzheimer's disease, they harbor two proteins in their brain that have changed, aggregated, and we can see a death. One is amyloid plaques. We now have imaging methods for that. We have ways to detect that in cerebrospinal fluid. And now we can also detect it in blood, and I'll tell you more about that later. The other and potentially more toxic protein is called tau. Both of these proteins are expressed in everyone's brain. They actually have a function. They're quite important. But in state of disease, they change their structure. It's also potentially because our brain is no longer clearing things from the brain. So why they accumulate in the brain, I think, is our clue as to why we develop neurodegenerative diseases. I consider them just to be biomarkers of Alzheimer's disease. They mark the state of disease. They don't tell us why a brain went from being normal to having neurodegenerative disease. Now, we have some treatments against Alzheimer's neuropathology, and I'll show you in a future slide how they work. But really, we are detecting disease too late. So if we recognize disease when someone has full-blown symptoms, and if we only measure these two proteins, amyloid and tau, we're going for the tip of the iceberg. We have lived an entire life. We're a lot more than just the tip of the iceberg. And so the data that we are collecting globally now in research is much more than that. You're going to hear from Mike Snyder later today about how the use of wearables can transform many disorders. That is coming to neurodegenerative diseases and dementia. Imagine that you don't need to wait to develop full-blown Parkinson's disease, but you have a wearable that tells you that you're at higher risk because your movement has changed in a subtle way. You may feel it, others may not see it yet. That's the time where you want to detect the abnormality. Imagine that you wear, again, a wearable, an easy device, and you know that your sleep is now fragmented and that you need to do something about your sleep. And imagine that you give a blood test to measure your cholesterol or glucose levels and check your liver function, kidney function, and you also get to check your brain health. This is the data that is being generated with exponential speed in research. So we've gone from 1 to 2 to 3 to thousands of molecules that we're measuring in someone's blood, just microliters, a tiny amount of blood, we can measure thousands of molecules. Instead of waiting for someone to no longer be able to function in their daily life, we are measuring traits, abnormalities for that person. So change within the person, not on a group level or average. We then, you've heard about AI and you're going to hear more, we apply machine learning algorithms. We don't look at one hypothesis at a time, one molecule, one outcome. We throw the kitchen sink of data and we apply advanced analytical methods to see where the key abnormalities are. Doing this, we know that there is not one kind of Alzheimer's disease or one kind of dementia, but I put 30, but potentially hundreds. We all degenerate in different ways, and the treatments for each disease or disorder may be different. And what's most exciting, and this is a big focus for my lab, is that we're seeing connections between the body and the brain. This opens a whole new avenue for therapeutics. Imagine that you don't need to actually deliver treatments to the brain, but that you get to treat pathology in the body. You've heard about the gut-brain axis, but there's a lot more to that than just gut-brain axis. So over the past decade, maybe decades, but really technology has advanced to a great extent over the past decade, we get to connect the human genome, the genes, to the exposures that we go through. We can record that. And I hope that we'll do it more so and at scale. And we get to measure molecules in a non-invasive way. We don't need to wait for someone to die or to do an invasive lumbar puncture to get cerebrospinal fluid. We get to collect blood. And technology has gotten so good that we get to measure thousands of molecules in someone's blood. So in a cartoon fashion, the rich data that we have gathered over the past several years is showing us there's a lot more connection between diseases that have been siloed with brain degenerative disease, including Alzheimer's disease, than we thought there were. This provides incredible opportunities for drug repurposing, and there have been some trials. The problem with those trials is not that the concept is wrong, it's that we are not selecting individuals correctly for the trials. It's not going to be every single person who has Alzheimer's disease who may have a risk for diabetes or hypertension. Biology is very diverse. Again, many different roads lead to Alzheimer's disease, and we ought to use our precision methods in order to select the right group of individuals to test the right drug, and that's coming. So not one Alzheimer's disease, and in fact, not one neurodegenerative disease, but many. And what they do share is how we've come to define them. This is pretty arbitrary. We're saying that amyloid and tau need to be in the brain, but when we look at the brain of an individual with Alzheimer's disease, we see a lot more than that. So this is the big paper from one of the clinical trials going after amyloid. You may have heard about this drug, lecanemab. There are many more mAbs against amyloid that are coming to the market and are getting FDA approval. So this is one class of drugs that we're going to have at our disposal. It is going for the tip of the iceberg, but even going for the tip of the iceberg, you have an effect. You have an effect. So on the left, I'm showing you the biomarker. This is measuring amyloid in the brain using PET scans and imaging method, and showing that people who get drug clear the brain of amyloid. Great. The figure in the middle is showing you clinical improvement, meaning that people who get drug decline less fast by a bit. So you get completely rid of the pathology, but you hit the clinical symptom just a bit. A bit is better than nothing, but a bit is not ending dementia. So how we imagine the future as a field is that it's going to take many drugs. And I would say that if we go early enough, it's going to be fewer interventions because disease is less complicated. That's just logical. So by going for the tip of the iceberg, I find that actually quite encouraging. I thought that none of these drugs would work because it's really the tip of the iceberg. It's a lot that has led a brain to accumulate amyloid, and yet we did have some improvement. Imagine that you can measure many different pathologies with great precision early enough. You can do a lot more than just slow decline by a bit. I think you can slow decline to the point that cardiologists need to get on their feet and develop new drugs because the heart is going to fail or other organs are going to fail. At this point, the brain is failing when someone dies of dementia. By a show of hands, if we had a really good blood marker to detect neurodegenerative diseases and Alzheimer's disease, who would have their blood, who would have that measured? Fantastic. We are worried about this as a field. We are slow at bringing these innovations to the clinic. We have a blood test. We have a really fantastic blood test. What we keep on worrying about, and by we, not me, but our field keeps on worrying about, is giving someone a really heavy diagnosis with which they can't do anything about. And I think that the drug that I just showed you is not game-changing, but it's giving us hope. We know that if we detect disease at a symptomatic time point, we can do something about it. Imagine that we detect disease at an asymptomatic time point, meaning that your function is completely preserved, you're normal. But on the blood test, you are at risk of developing Alzheimer's disease in the future or neurodegenerative diseases of other kind. It's really wonderful to see that you would do that blood test because that's the way to go. You have a chance at that point to modify your life in ways that you could potentially mitigate this risk. This is what we've done for heart disease. This is what we're doing for cancer. You detect cancer in situ. It's abnormal cells only in one region of the brain, and you can do something about that. You don't wait until cancer is metastatic before you go after it. This is going to be absolutely game-changing, and it's very simple. We just need a technology to be able to measure these molecules with great precision in blood. Now, how we're going to use it, I don't think is going to be as game-changing, but it's coming. Here I'm showing you the red, the yellow, and the blue, and we're basically dividing people up. And we're saying if you're in the red zone, you definitely have, or most definitely have, Alzheimer's disease pathology. If you're in the yellow zone, we really don't know. You could or you could not. And if you're in the blue zone, you can breathe easier, you don't have it. But this is wrong. We're bucketing people. We shouldn't be bucketing something that we all will develop if we live long enough. It's not about putting people in buckets and potentially having some sort of stigma associated with those buckets. It's a continuum. How we should be using these blood tests is that we all get it as part of an annual, a biannual. Same level as measuring your cholesterol or glucose or other organ function, and then looking to see where you're trending. Are you going in the right direction? Are you staying steady cold? Or are you improving those levels? This is how we should be using these blood tests, because this is really what they represent. They're actually very, very dynamic. So I think very exciting to have these blood tests and to be able to screen. But what I'm most excited about and what my lab is doing is, alongside these 2, 3, 4 markers of brain health or degeneration, we measure thousands of proteins in someone's blood, and tiny amounts of blood are needed in order to do that. What that shows us are perturbations in someone's physiology and biology. And what we do, because not every perturbation is pathologic or abnormal, we then look at that person's function. And we do that not by quantifying memory impairment or cognitive decline or inability to perform tasks of daily living, but we measure things like processing speed. Processing speed declines with aging. We measure someone's speed of gait. We measure the quality of their sleep. We are really interested in functions that alter with aging, and that may alter more so when someone develops a neurodegenerative disease, but still early, before irreparable damage has been done. When we do that, we uncover biology. When we uncover biology, we want to understand mechanism. How we get a mechanism is not by turning to mouse models or other kinds of animal models. They're useful once you have a drug and you want to show that they're not toxic, but they don't represent humans, and they definitely don't represent the complicated functions that get altered with neurodegenerative disease. So what we do is we derive stem cells from blood donations, and we develop, you heard about organoids earlier on, we develop cells, primary cells of human brain. We are really interested in vasculature, but we can also do this for brain tissue, and then we screen drug against that. This is how we conquered some cancers, and this is how we're going to conquer some of the neurodegenerative disorders. Here I'm showing you a plot where we just measure proteins, 80 proteins in people's blood, and we get to put them in biological categories. ApoE4 is one of the most prevalent risk factors for Alzheimer's disease, and here I'm showing you individuals with Alzheimer's disease carrying ApoE4, no Alzheimer's disease carrying ApoE4, and the control groups all cluster in different groups. No age, no sex, no demographic, no cognitive function was entered into the machine learning algorithm in this case, only protein level. So 80 proteins in blood get to separate people. What this tells us is that there's a signature in people's blood that we can go after, and that is related to biology. What I am obsessed with is not the brain tissue, is the blood vessels of the brain. We're born with our neurons, and they are with us all our life. They're minding their own business and they're functioning. What gets exposed to our environment and changes all the time on a daily basis is blood. That's the content of your lived experiences. And that's what the brain sees. However, the brain is protected from the environment through a barrier called the blood-brain barrier. This changes with age, and we think that this is a key component to all neurodegenerative diseases. And what we want to do is understand what are the changes that put the brain at risk of degeneration and go after those with therapeutics. The advantage of going for the barriers of the brain is also the lack of trouble for delivering the drug to the brain. We just need to put it in the bloodstream and it'll go where it needs to go. Ultimately, everything that my lab is doing at this point is to develop methods to detect early and reduce the possibilities of pathology down to what is most therapeutically relevant and treatable for a given individual. It's not going to be a one-size-fits-all, and we want to be able to detect with great precision what we need to treat in whom. This is what we want. All roads are leading to dementia and neurodegenerative diseases, and we want to be able to tell someone that your cholesterol level needs to be here. This is what the blood pressure is. It's not 120 systolic for everyone. For you, it needs to be 10 or 100 systolic, and you need to sleep these many hours, you need to do this, you need to eat this. This is within the realm of possibility. It's just that we have gone for group level therapeutics. It's good, it's a good start, but we now have techniques and tools to be more precise. And this is the dream pathway. You have early detection of clinical abnormalities, you subtype the disease using biomarkers, you intervene, you normalize symptoms. Or you go even earlier. You look at someone's genome, you know their environment, you use biomarkers to detect risk, you intervene, you normalize the biomarkers. Function has not even altered. I think we need a complete paradigm shift if that's what we're gonna do, because people are gonna say there are ethical barriers. You can't diagnose someone who's totally normal with a disease, and I would say that you can if you have the biological markers of disease, and that's exactly when we need to diagnose disease. And ultimately in my clinic, this is what I want to do. When someone walks in and says, I can't do this or that, or I feel funny before anyone else is realizing that there's something off, I want to have a whole panel of biomarkers that I can run and tell them that the blood vessels in your brain are dysfunctional in this way and that way, or that they're not, they're okay, and the abnormalities are a different kind, and have the choice of treatment for that individual. We're not actually far off. Growth of our knowledge has really been exponential, and this is going to be the future. So we've had a very productive decade. We are going from 1 or 2 molecules to thousands of molecules that we can measure. We're going for traits, for abnormalities that are well before someone's function can decline. And we can do this with unprecedented precision in research. We have not brought it to clinic, but I think it's time to start pushing the boundaries and bring the technology that we have at our disposal in research to clinic. All of this is going to lead to new developments in therapeutics, and this is what's going to be really game-changing. What we need to do is tailor treatments, and the earlier you go, the more tailored the treatment can be, and the less toxic the treatment can be. What we want to do is establish balance. What I haven't told you about is the incredible resilience of the brain. The fact that we all die with amount of pathology, and not everyone actually has symptoms, that's because our brain is really resilient. Our brain is key to many of our human functions, and so the biology of resilience has really been selected for. Thank you for your attention.